didn't sit and working, I hope you don't mind. And I will try to answer two questions. The first one was about if we can compare data from different experiments, from totally different studies, and how we can do that. And the second question was about the formulating of biological questions, and are there any ways to do it on a more broad level? It was your question. Yeah. And I will start with very basic stuff. For me, this picture is kind of very interesting, but probably any biologist in the university studied this, the central dogma of molecular biology. And these two gentlemen are very well known. They discovered double-headed DNA structure, and also in 1970, Francis Crick also formulated the central dogma, and he basically described how the information flow in a cell. And we start with DNA, which can copy information through the replication, that the information is transcribed to RNA, and finally translated to the protein level. And it was formulated in a way that it is uniflow, move all the information, and we don't have any backward stuff going on. And they probably, we can distribute the slides later, because I try to give a reference on all the stuff from these slides. And for example, this was from Han Academy, nice videos which helped me at some point of time. And sorry for all this basic stuff, but I would like you to think about the information in the cell, in DNA, RNA, in a more like a mathematical problem or a programmatical. If any of you ever tried to finish some bioinformatics courses, probably one of the first tasks is to implement the function. Even, for example, your string and so on, you are asked to implement the function which converts your string to a complementary string, or for example to a reverse complementary string. And you can do it through the cycling of these bases, and you can do it even in a parallel manner if you try to divide your string in different chunks and give these chunks to different CPU, and then you can concatenate the results. So, I mean, for me it's like this, not like in the first lecture probably. That's why I was really fascinated when listening to all the details of gene expression. And the same actually goes for protein level, but in this case we have a finite number of states, because we have a finite number of amino acids, and we don't need probably such kind of function, but we can define all the transition states. We can put it in database and basically search this database and got the answer how to translate amino acid into the codons. But people have been struggling to decipher this information, these genetic codes, for years actually. And probably a lot of you know about Human Genome Project. It started in 1990, and it took 3 years to actually have this reference genome, which was in the end comprised from several genomes, so you can say that this is some kind of an average genome. And so the goal was just to decipher DNA sequence and identify and map all the genes. and actually find the functions of the genes. And what was in the end, first of all, it took almost $3 billion to finish this, so it's very expensive. And they found up to 20,000 genes which code the proteins. And after finishing this project, another project started just right away. And why? So the previous results is estimated that humans have like this 20,000 protein coding genes, but it's almost like 2% of DNA in the human genome. So people really used to think that the rest is a job, but it's not like that. And this Encyclopedia of DNA Elements project had also very important implications, and they studied the functions of non-coding RNA molecules and so on. It was actually, I think, mentioned on the first lecture. And if you remember, the Human Genome Project started in 1990, and it's so the first records they got in 2003. So unfortunately, they were doing it using the old sequencing technologies, and that's why it took them like 13 years. But then, if I remember, it was 2007, Illumina introduced its sequencer to the market, and it started the new era of which we call the next generation sequencing. Now it's a very fancy type of research, which is single molecular sequencing, and people even expecting new interesting things. If probably some of you heard about like small pocket sequencer, which look like a flash drive, and people even sequencing some stuff in the space using this. So people are doing a lot of amazing things we can only dream about, unfortunately. And I hope Roma is going to show this slide, because this is something very, very typical. And it is about DNA sequences cost per genome. As you remember, the Human Genome Project costed $3 billion, and now we can actually do it not for 13 years, but for a few weeks and for a very low amount of money in comparison to what people did before. And as a result, we now have a lot of different assays. I would like you to visit this link. It's a blog of very known researcher Leor Pachter. He is actually an author of this new ultra-fast mapping tool Callisto, probably Roma knows. And on this page, he actually tries to annotate all the seq assays he find in the literature, and now he has up to 100 different assays. Some of these names I don't even know, and I don't know what they are doing. Well, of course we know what is mass spectrometry, I don't know, Hi-C, but a lot of data is being generated from the so-called omics studies. So genomics or genomics has a suffix omics. And now we have this multi-omics concept. On the genome level, we can see what can happen. In the transcriptome level, what appears to be happening. On the proteome, what has happened and what makes it happen. And metabolome, what has happened and what is happening. But what can we say on the more broad level? On the level of cell and organism, we are still unable to mathematically formulate this kind of complexity, even though there are some projects that tries to model a cell, they are far from to be perfect. So, and the question, what kind of science should study this kind of level of complexity? And before we answer the question, which probably obvious from the name of my presentation, I would like you to look on the expanded central dogma of molecular biology. And so this slide was taken from a very nice paper. And that paper is about the emerging of new field which is called editor tone. And they try to formulate their view of expanded dogma and what is necessary. Now we can say that content can flow backwards or even sideways. And the information also can be inherited or transmitted, like, I don't know, through a viral RNA and so on. So for a mathematician or for a programmer, this picture is much more complicated than the first one. That's why probably we need new approaches, more complex algorithms to try to solve all this stuff and to try to give a model which describes all these things simultaneously. Well, while the next generation sequences technologies were emerging, another interesting events took place, and they are more about mathematical modeling of biological processes done usually by mathematicians, sometimes with a very naive visualization what they think biological processes are, but still they tried to apply their mathematical stuff to biological problems, but it was not very widespread probably until the next generation sequences technologies emerged, which actually, so these two branches of events gave birth to systems analysis, which was critical to molecular biology in general. And finally, in 2000, the first Institute for Systems Biology was established by Larry Hood. So this branch of science is, I would say, very young, and it's a quotation of Larry Hood. So the whole trinity of system biology is biology, computational science, and technology. And of course, it's so information flow in this case is constantly cycling. We are getting new data. We try to formulate hypotheses, and we ask people to generate more data to support or to decline our hypothesis. So what is systems biology? So first of all, it's quantitative analysis, then it's holistic approach, contrary to reductionist approach, and the systems biology aims at understanding the behavior of the system as a whole, not only the parts of the system. Well, this is a little bit, I would say, not very precise, because sometimes it's very hard to distinguish between different fields of science. You cannot separate one from another, but still, anyway, this thing actually funny that systems biology initially gathers data without prior hypothesis, like let's sequence this, let's generate a lot of data, and then we will try to see if we have something in that. Well, and actually it is happening in a lot of cases, especially, I don't know, in China. They would like to sequence a lot of things, but I would say it's more or less true. But it's what I like about the systems biology, because from the same set of data, you can try to infer different things. We could clearly see it from the Roma's talk, because if you want to look on absolute abundances or relative, you could solve totally different problems. And the systems biology study properties of biological systems, obviously. And what I would like to point here is this one. So the so-called emergent properties, which you cannot derive from separate parts of the system, and these properties appears only when the parts are combined and you look on this whole level. This is very important because it plays a key role in understanding what is systems biology and what are the tools of the systems biology. And actually the central idea is reverse engineering. I will tell what is it on the next slide. And here you can see that actually systems biology try to use successful findings from all other disciplines, physics, mathematics, and so on. A lot of machine learning have been applied, or particular, for example, like information theory approaches, which are based on Shannon entropy definition. So sometimes you can be even lost in this variety of methods. That's why I'm really grateful when someone can share the knowledge and tell me, for example, use this tool and don't use this tool. And what does the reverse engineer mean? Consider we have our matrix of values. It can be from RNA sequencing or from microarrays. It doesn't matter in this case, but we have our genes, and for example in the columns and in the rows we can have different samples, or we can have different states, so it's basically a condition. And then we do this matrix of numbers. We can apply different types of analysis, different types of inference. We can use, I don't know, simple correlation. And in the end, we will get such kind of model in a graph representation. So if X, Y, Z are genes, then the edges on this graph represent the relationship between these genes. And finally, the main point of this talk is about data actually integration. And what I want to say that it can be performed on two different levels. First is data integration, which is on this level. And second is models integration, which can happen on this level. Because you cannot always obtain the raw data or the numeric data. Sometimes you can just get this kind of thing from someone's paper or from database. So we need both kind of methods to solve the problems. And the first one, or data integration, can be divided in two subtopics. First is integration within similar data types. For example, if we have two RNA sequencing studies, one is 20 samples and the other one is 15 samples, and you really would like to look onto the differentially expressed genes. So your question is what to do with these two matrices of values. And the second one is integration between different experimental data is when, for example, you have RNA sequences data and, I don't know, copy number variations or methylation data, and you would like to connect two of these different processes into the one model. So let's look on these things first, and the data integration with similar types of experiments. First, we need to understand where we can get the data, and I usually use NCBI databases like Gene Expression Omnibus and Sequence Reader Archive, and the other concurrent database is RAExpress, and actually the most of studies are present in both databases. They have two identifiers. And the third one is about proteomics data. Actually, NCBI had the protein assays, but unfortunately they had to discontinue this database because of lack of money. Even such kind of big consortia institution, they sometimes cannot afford supporting these public repositories. So now this is a chart from a Gene Expression Omnibus and CBI database, and actually basically it's distribution of the number and types of selected studies per each year, and we can see that here is our expression profiling by next generation sequencing, and it's to answer the question why people still study microarrays, because the number of data made with that arrays are much more bigger than the next generation sequencing still. So you can have a lot of choices in choosing your data, even if you cannot do it in your laboratory. So in terms of integrating data within the same type of experiment, we have again two options. The first one is meta-analysis, so the results of individual studies are combined at the interpretative level. And the second approach is merging. So basically you need to adjust your, for example, expression values level in such manner that they can be comparable directly and numerically. And in the meta-analysis technique, the most common situation is the 2+ comparison. For example, we have experiment and control, and where the objective is to identify genes expressed differentially, or the transcripts of which are being produced at significantly lower or higher numbers. And this is a typical heat map, for we have here one condition and the second one, and this is a list of differentially expressed genes, and you can see that the red marks are for more highly expressed genes, and the blue one is low expressed genes. And let's say we've got several lists of differentially expressed genes. One, for example, you obtained during your own experiment, and the second list, for example, you obtained from some article which you are interested in. And actually, this is a slide. I'm not going to go deep into the different types of methods which try to find differentially expressed genes, but I just wanted to give you this citation so you can look later. But the step of choosing the methods for differentially expressed genes analysis also matters. And these are the charts for the comparison of such methods for RNA sequencing. And you can see that they can perform very, very differently, especially when it comes to low expressed genes. So, again, we have several lists of differentially expressed genes which came from different studies. So what can we do with that? First, it's vote counting, then we can combine ranks, we can try to combine p-values or effect sizes. I will tell you a little bit more about the first two because it's really easy to explain. And in case of what counting methods, so you compute statistical significance of differential expression, so you got your list with p-values, and per each gene, count the number of studies that have p-values smaller than the predefined threshold. For example, also 0.05. And let's say, for example, the third one is genes with what count more than a predefined threshold. For example, you have 10 studies and you decided that your threshold is 5, then you expect this gene of interest to appear at least in 5 studies. So if the gene does appear, you can say that, okay, I will count it as really, really differentially expressed. The second type is rank aggregation, and here we can actually formulate it as an optimization problem, and our objective is to find a superlist which would be as close as possible to all individual ordered lists simultaneously. If you're interested, you can actually try this R package, which is called rank aggregation, and it implements the following algorithms, but I will even try to explain that. Sorry for the formulas, but actually it's very, very easy. So let's say we have first list with the 4 genes like A, B, C, D, and the second gene is, for example, B, C, the second list, B, C, D, A. So, what is the rank of gene A in this list? Yes. And in this list, 4. And let's say we try to find a list which is as close to this list and to this list as possible, and at the same times. For this, we can try to develop some kind of distance function between this list and this and this and this. And we can actually take a very, very simple distance function, which is the subtraction of the rank from one list and the second one. Let's say, for example, let's find the distance of gene A between this list and this one. So this is basically distance of gene A in list 1 minus distance in list 2 and modulo. So we don't want the negative values. So it's kind of very easy. We have 1 minus 4, which is 3, not minus. Here this is the mathematical way how you can obtain the positive values. So 3. So in our process, we try to find all the distances of all the genes between all the lists, and we try to actually minimize this function which can be solved using, for example, regression analysis, like simple linear regression. I'm not going to tell you about this, but you don't have to implement it yourself. You can go with any R package. But if you would really want to understand it, I recommend machine learning course on Coursera, which is very, very amazing, and it is almost about regression analysis. So from one point of view it looks like scary, but actually if you take a piece of paper you can solve this by hand if you have small lists and you have small number of genes.
And what does this distance mean? Is it difference in expression which you're trying to get closer between two lists?
First, let's say you have actually, it can be solved in different ways. For example, when you get your list of differentially expressed genes between two conditions, you will get first of all a p-value, and then you will get, for example, logarithmic fold change between conditions. And normally you would first sort based on this stuff, and only that you will try to sort based on this stuff. And for example, let's say that gene A has the biggest fold change. That's why we give rank number 1 to gene A. So it is connected with expression, but it's kind of course, a simplification because we need ways how to compare it between different experiments, because we cannot compare it numerically. So we'll try to compare it in a sense of ranking.
So because you cannot compare it by fold nor by p-value, you will rank each gene in these two data sets and then compare the ranking.
Yes.
And if they compare, the rankings compare, then you can compare the fold change.
No,
not.
it's not like that. The task is formulated like this. Again, let's say we have 10 lists of differentially expressed genes from 10 studies. But your question, how is significant that gene A is very, very bright gene? But because, for example, in one list you have it ranked first, and in all other lists you have it ranked the last. So in this case, you are trying to give a gene a position in your list which in accordance of all your lists which you obtained from different experiments. And then, for example, when you get this super list, and it can be, I don't know, hundreds, even thousands of genes, but you will have your gene of important, for example, top 20 genes. And you will be at least a little bit more sure that these top 20 are supported by different experiments, not only by the one.
So the lower the distance, the more it is probable that the gene in question is as expressed as you think it is, right? And if the distance is huge, then there is different values in different studies, and you're not so sure, right?
More or less, yes, but actually don't think that distance here is something very defined. I mean, you can use any distance function. I show this one because it's very easy to understand. But for example, you can actually, here you are trying to combine weighted lists. For example, for some reason you think that the data from this study is more trustworthy, so you will give it a little bit more of a weight, just in terms of constant coefficient. And I don't know, your distance function can be a lot more sophisticated with, I don't know, logarithms or exponents and so on. So it really depends on the study or on your mathematical understanding of how to compare these rankings. But if, for example, we have the gene A ranked first in all the analysis, yes, we are more sure that this gene of a significant importance. And if we have some gene, for example, B, jumping from one position to another, probably we shouldn't consider it when trying to solve a particular problem, because different studies show the different position of the gene.
My last question. Maybe it's normal to be in different places. Maybe it's variable.
Yes, but if you want to tell difference between two conditions, for example, control experiment, and you would like to, for example, infer gene signatures which are strongly associated with, I don't know, some disease, you would not want some jumping gene. It might be normal. I mean, it depends on the formulated problem. Okay, so that was the previously we discussed the situation when we cannot directly compare the numbers, but in some cases we actually can, and but you need to differentiate between two kinds of normalization. The first one is within study normalization. For example, Roma showed us RPK and TPM metrics, and they kind of used to normalize RNA-seq data, and actually this one, RGB, it's remove unwanted variation, and as far as I remember, it utilizes this spiking stuff you were talking about. And there are different methods for microarrays and for RNA-seq, and since we are talking here more about transcriptomic level, I try to make a focus on this stuff. And so if you have a data set which was produced during one experiment, you would normally want to normalize it within your study. But the other very important aspect is the cross-study normalization, which aims to remove so-called batch effect, which is, in a simple words, a technical variation, and it can appear for sometimes even for unknown reasons, but usually it happens if you are doing your experiment within some time frame. For example, you did this bunch of sample this month, and the second bunch of sample you did the next month, so you are expect to have a batch effect. This is the illustration what the batch effect is, and to make this plot, I took the data from this paper. So they basically uploaded the microarray data to gene expression omnibus, and you can obtain the raw values, and you can do anything you want with them. You can even try to replicate what I did. So this is the principal component analysis plot, and each dot here represents the one sample. So even though each sample has, I don't know, up to 20,000 genes, we are able to reduce the dimensionality of our data and have a two-dimensional plot. Lucky for me, in this article, they actually stated they have two batches. So they knew they would have this technical variation, but it's not always the case. Even now, you can get the paper, the results of which were based on the false data analysis, which is kind of sad because this thing is very well known. And after the batch effect correction, you see that the dots are more sparse, and you can actually kind of see that they can cluster. So the red ones are control and the blue ones are experiments. So we can draw, for example, line here, and I don't know, build a classificator which will assign a class to all the dots which are above the line, and another class to all the dots which are below the line. And for this example, I used combat method from SVR package, which I highly recommend, because actually, even though this example are based on microarray data, people also use this combat method for adjusting batch effect for RNA sequencing data as well. And once I had a question about this plot, because the question was, how do you actually know that this is a batch? Maybe this is some hidden biological variable. That's why first it is important to get as much data about your sample as you can, not only expression values but any clinical data, so you can test these additional variables if they correlate with these two different classes. And the second way how you can try to tell if this is the technical batch effect, to know. Usually when you measure when you get your expression profile, you expect that there are a lot of different processes happening in the cell at the same time. So probably your samples won't be so similar. So here you see like broad, scarce plot, and this one is a naturally lined along one axis. It's not happening when you have, so you have your individual differences between samples, and this looks like they have a natural way of lining. So from the experience, usually when you see such plots pretty oftenly, you can just tell by looking that this data has batch effect and this doesn't. And there are some actually interesting sites, like I know one for microarrays and another one for RNA-seq data, and they collect like typical artifacts, typical errors, which comes from wrong library preparation, I don't know, wrong data analysis. So you can try to look and find a similar picture to try to understand if your data analysis is correct. So, but the question is, which way is better? Should we use meta-analysis or direct data integration? Do we get more statistically significant results? And to illustrate you, so this method looks like very nice, but to illustrate you the problems in this method, I will show you this plot. So this plot I made using 3 different data sets, again from gene expression omnibus, and 2 of the data sets were made using the same microarray chip of the same version, and the 3rd data set was made by a newer version of an Affymetrix chip. All the 3 data sets, they use samples which come from different patients. But even though if you compare the data made on the same chip version, you can see that actually the correlation is pretty high. Here, each dot represents a gene, and on axes we have log transformed process intensity values, or you can tell these gene expression levels. So this is synonymical in this case. And I built 2 lines of regression both linear, but one is using linear function, and the blue one is actually a cubic function. And here you see the comparison of older version of photometric shift and newer version. And the correlation significantly drops. And when you see such kind of picture, you ask yourself a question whether they actually can try to combine expression values into one matrix if the data are so influenced by the technology you use. But surprisingly, it is still better. There are some studies that show that if you combine different data sets, you will get much more of differentially expressed genes. And if we say that, for example, RNA sequencing data are much more precise, and we use it as a reference, and we compare it with the microarray data, and if the integrated data set correlates better with RNA-seq, it at least can identify that the integrating approach is not worse than meta-analysis. But anyway, when you try to choose your tool, you need to understand what kind of data you can operate with, because no matter what kind of the analysis you want to do, I will always recommend you to start with the raw data, because the initial authors might have missed this batch effect, and you will have to correct it yourself. So don't trust people. Even more complex task is to try to integrate data between experiments of different types, so-called multi-omics approaches. So this is to remind you that sometimes we can deal with broad diversity of data types. Unfortunately, it's a little bit not seen, but I would recommend reading this article, because it is actually about multi-omics data integration. And I don't know, you can even try to integrate all these types of data, but of course you need to be very careful to choose your methods. And these kind of approaches have their own drawbacks, like a restricted availability of multi-omics data sets for the same samples, because normally you would want your methylation data and RNA data to come from the same individual, but it's not always possible, and probably it's not always necessary. It again depends on how you formulate your problem. Again, while some types of experiment give us a lot of measured variables, in other ones, I don't know, like proteomics or metabolomics, they give us a number of information on a smaller degree, and it's not always straightforward to try to combine the data of a bigger scale with a smaller one. And again, the big problem is that biological processes are happening in time, and the data flow on different levels are very different. Like, I don't know, transcriptomic and metabolic, totally different speed of reactions and so on. So I would also recommend you to read this review. I'm not going to cover all these tools, but they are mostly about multi-omics studies, and the author tried to give a little bit different categorization of the tools than I am trying to show you. And for example, he grouped the tools to those who try to study molecular mechanism, we try to define sample clustering, because not always you will get the situation when you have your control cohort and your experiment cohort well defined. For example, you would want to study cancer subtypes, and you think that all other researchers did it in a wrong way, and you want to make your own subtypes. That's why you usually have just, if we go with this plot, imagine that you don't have this coloring, you only have the dots. And you want some algorithm that will tell you that this part is class 1, and this part is class 2. And you will say that this is a cancer subtype 1, and you need to treat the patient of this cancer type with the medicine of particular type, and this cluster of patients should be treated totally differently. So this is a very important question, which basically depends on people life. So the majority of articles about cancer studies are about gene signatures inference. I don't have this citation, but funny that there is an article which basically proved that all these gene signatures are not very relevant, because they were randomly generating gene lists, and they were as much associated with the patient's I don't know, cancer class or outcome as the gene signatures are reported in the majority of papers. And the third class is prediction. Again, if you study cancer, you want to try to predict whether this patient survived. Sometimes you predict that he or she is not, which is actually very, very sad. And another formulas, I'm so sorry, but I will try to explain this as well. And in this case, we have 3 different types of data: gene expression, DNA methylation, and copy number variation data. And let's say we came up with some kind of formula which calculates score for each gene, and this score depends on the results of DNA methylation and C and V data. So basically, I will come back to the slide, but I want to show you the steps of this algorithm. So we will try to divide our methylation data to normal and hypermethylated, or normal and hypermethylated samples. And we indicate these samples by 0 or 1 in our label matrix. It will be like this. Let's say we have sample 1, sample 2, and so on. And here we have gene 1, gene 2, and so on to gene N. And we will also add additional data to this matrix, for example, methylation rate, and we will assign 0 or 1 to our samples, depending on what we decide, if the sample hypomethylated or hyper, and the same with CNA data. We have the 0, 0, 1, and so on. And basically we expand our metrics of data and we present our knowledge about population and CNV in a very, very simple way, just 0 or 1. And we will try to calculate our score based on the mean differences and variation differences of samples which have the zeros and samples which have ones. You can compare it with differentially expressed genes analysis, but instead of conditions, well, here we also have a condition, it's 0 or 1, but we assign this condition not from clinical data, like this patient is ill and this one is healthy. We try to assign this class based on other NGS data we have. So once again, so for each gene we calculate this term, and then we we calculate separately for methylation and separately for copy number variation. Then we try to sum this one and multiply by some error measure. And then we sum two statistics together, and in the end we will get the score per each gene, which would be associated both simultaneously with CNV data and methylation data, and we then can, for example, work with expression values. So this, you can imagine, it's also as some kind of a filtering technique, a little different to differential analysis. This is a totally another study, and so the major difference between the previous one and this one is that they actually combined all the data matrices. First, they had 3 different data sets of mRNA, and they chose only those genes that are correlated between these matrices. So they filtered these genes, and then they combined the matrices, and finally they added microRNA and protein assays. So they got a very, very huge matrix of numbers, if I were to describe that. So this joint matrix, and they then applied such a technique as factor analysis, which I try to describe on this picture. In a way, it is similar to principal component analysis, which I showed you before, but with few differences. Actually, the PCA is a subclass of a more broad class of factor analysis methods, and the main differences are that in case of PCA, they have our features or genes or here items, it doesn't matter how you call them, and we reduce them into components. For example, so we have principal component 1, which, for example, explains 9% of diversity of genes, and we have principal component 2, which explains 7% of diversity of genes, and so on and so on. And the number of principal components is less or equals to the number of all genes. And this is okay. I will try to be faster. Okay, the difference with factor analysis that we suppose that there is some hidden underlying factor that influence your gene, and also we try to add to this model some kind of measurement error. But in other techniques, these two methods are pretty much similar, especially in terms of what kind of mathematical stuff you need to apply to actually get your principal components or factors. Here we have again a simple formula where X is our omics joint matrix, and we have these hidden factors, and we want to find, and basically we again can apply or linear algebra matrix operations, or we can try to apply linear regression to solve this equation. And finally, you can get your factors, but again, for example, you got 100 factors, but still you need to filter those factors and choose only factors which are associated, for example, with cancer cell lines, or with the patient outcomes, or with the medicine you give to the patient, and so on and so on. So this I'm going to skip. So basically the most important sentence is this. So they compared the approach to the separate analysis of separate studies, and they showed that the joint matrix and their factors are more associated with their biological variables information than when you study each data set separately. And the last one, I will try to be very fast, is models integration, where the biological networks can be represented as graphs where vertices are genes and edges, for example, interaction between genes. And you can actually obtain these biological networks, for example, analytically. You can combine different information you can get from different databases, or you can do data mining using a vast number of scientific articles. And the second type of approaches is the regulatory network inferences. So you can have a different representation of biological networks, just a simple graph, some Bayesian network or Boolean network, and the inference methods are dependent on the way how you represent this. But this is a very difficult stuff, because usually the very naive approaches which try to brute force finding the optimal network, they are very slow, and if you use methods which are not precise, we have some restriction, they're called heuristics methods, you will have a more accurate model. And there are number of techniques you can try to study your network, and then you can even participate in some challenges to try your method, and then you can try to apply networks alignment. This is a totally different types of tools. And the last slide, I would recommend you these two different tools. First one is Satyascape, and the second one BioLayout. There you can try a lot of different plugins, a lot of different types of representation to try to work with your biological data on the networks level. And I think we'll skip this.
No! That's the conclusion.
Okay. Basically, I think that even though I tried to show you a variety of different tools, personally I'm not satisfied with a majority of them. For example, these two tools, I hate them because they are implemented in Java and they are so slow. And if I'm trying to upload my network, which has for at least 100 connections, it becomes practically impossible to use them. And I truly think that people from different fields of sciences should cooperate, like programmers, biologists, and mathematicians. Because Still, I don't believe in end-user solutions. I'm sorry, but at least half-end solutions should be possible, but we don't even have these kind of tools. I mean, in systems biology, it is very, very different.